How's everybody doing? Nice, long 3-day weekend? 5-day weekend from biochemistry? Yeah. We have an exam coming up. That's the good news. A chance to demonstrate how much you know, which is good. Today I'm going to finish up talking about oxidative phosphorylation and respiratory control, and they go hand in hand, as we shall see. There are some very interesting and cool things to think about with respect to those, and if I have time today, I'm also planning to talk about the photosynthetic fish, which is one of my most popular lectures. So I'll talk about that today if I can get to that, which I think I will. Well, last time we finished by talking about the rotation of the ATP synthase structure. And the ATP synthase structure looks something like this, looking from the top down. And we see the rotation of that inner core, that the rotation of the inner core changes the configuration of the surrounding proteins around it. And it's in those surrounding proteins that the ADP and the PI is bound, compressed, made into ATP, and released. And that is the way that we make about probably 95% of the ATP in our cells. Okay? This process is dependent upon a proton gradient. I can't emphasize that enough. It's dependent upon a proton gradient. A proton gradient requires an intact mitochondrial membrane. It requires electron transport. It requires oxygen, which is one of the things that electron transport depends upon. And if those conditions, and there's one other very important thing, ADP. Okay? We've been thinking a lot about how cells have to have NAD so they can make NADH, but you probably never thought about the fact that cells have to have ADP in order to make ATP. Right? They go back and forth. Right? If you have a lot of ATP, you have low ADP. Now that turns out to be a very important principle in understanding respiratory control that I'll be talking about today. So, intact mitochondrial membrane, right? Electron transport, oxygen, and ADP. Those four things, if we keep them in mind, we will understand respiratory control much better. Okay. Let's see. Don't think I need to mention that. And what can I say here? All this is doing is showing the movement of protons into the ATP synthase. They come in from the top, and coming in from the top, they load into this gamma chamber, which is the part that's in the middle, that's in the very middle of this thing. And it causes the gamma chamber to rotate, which in turn is causing the, what do I want to say? Causing the proteins around the gamma chamber. Gamma chamber is actually in the middle of this. It's a little confusing the way it looks here. But that causes the gamma chamber in the middle to rotate. As a result of the movement of the protons, the gamma chamber rotates and the ATP is synthesized. Yes? Okay, so his question is, do we actively make ADP or do we recycle a reservoir? Well, at some level, cells do have to make the precursors, so we'll talk a little bit about the making of nucleotides, just like they have to make NAD, right? So they have to make NAD as well, but they only make a fixed amount, and they also only make a fixed amount of ADP. So it's recycled on and on and on. I think I may have mentioned in this class before, an amazing statistic. If I didn't, I'll repeat it here because it still blows my mind. Every day, your body makes and breaks down its weight in ATP. Every day. Now we talk about a lot of ATP. That's a lot of ATP. Okay? You make and break down your body's weight in ATP every day. Okay. So that's oxidative phosphorylation. Right? It's oxidative because it's depending upon the oxidations and reductions that are happening in the electron transport system. Okay? One of the things to keep in mind with respect to the electron transport system is that we remember that NADH and FADH2 are the feeders of the system. They provide the electrons necessary for the electron transport system to go. One of the things that I haven't talked about, but I will tell you now, is that NADH does not cross the mitochondrial inner membrane. It does not cross it. Meaning that if you're making NADH out in the cytoplasm like you are with glycolysis, that NADH cannot directly come into the mitochondrial matrix. Okay? There are shuttles to get the electrons in. Shuttles. Okay? So what shuttles do is they use molecules that carry the electrons across the inner mitochondrial membrane, that can cross the membrane, and then when they get in, the electrons get donated to something to ultimately make NADH, or in some cases, FADH2. The simpler of these systems is the glycerol phosphate shuttle. It's depicted on the screen. It's very common in insect muscle. It's a simple system. And the beauty of this simple system is it works very rapidly. And it works as follows. If we look out of the cytoplasm, okay, we have dihydroxyacetone phosphate. That's a byproduct of glycolysis. Dihydroxyacetone phosphate can gain electrons and protons and become glycerol 3-phosphate. That's what's happening here. So we see NADH produced in glycolysis that now is becoming glycerol 3-phosphate. Why glycerol 3-phosphate? Because glycerol 3-phosphate can move its electrons through or into the mitochondrial inner membrane. It does it by donating its electrons to FAD, which is in a protein that's in the mitochondrial inner membrane. Very simple. Nothing has to move across the membrane. Things are moving into this protein that's in there. Consequently, the electrons make it into FAD, becomes FADH2, and dihydroxyacetone is reproduced. So we have a little cycle that's going on here. As I said, it's very simple. That's the good thing. The downside is it's inefficient. Because what we've done was we started with an NADH, and we ended up with an FADH2. FADH2, you remember, is through complex 2. And by the way, this is happening in complex 2. And that's what this yellow guy here is right here. This is happening in complex 2, and complex 2 bypasses complex 1, where protons get pumped. Now, you remember when we talked about the proton pumps, we said that there were protons pumped by electrons going through complex 1, complex 3, and complex 4. There's enough protons pumped from each one of those to make 1 ATP, ultimately. Okay? Enough protons get pumped through complexes 1, 3, and 4 to make 1 ATP each. That's a total of 3 ATPs for electrons that enter at complex 1. If something doesn't enter at complex 1, but instead enters at complex 2, by coming in through FADH2, we're only going to get 2 ATPs. Okay? So this is a rapid system, but it's inefficient. It doesn't pump as many protons as if the protons had come in through complex 1. To get things in through complex 1, we have to use a different shuttle system. The different shuttle system we use is called the Mele Aspartate shuttle. It looks a lot more complicated, but it's actually not nearly as complicated as it looks. In fact, I'm going to simplify it for you significantly.
Question?
Yes?
So you were saying that any NADH doesn't cross the membrane?
That's correct.
And FAD does?
So NADH does not cross the inner mitochondrial membrane. FADH2 does not either. It's going in through that complex 2. So it's not, nothing is crossing the membrane in what I've just shown you. You're just putting into a protein that happens to be in the membrane. Okay? Now, this system actually moves electrons across the membrane. This system is moving electrons across the membrane. Now, I'm not totally fond of this figure, to be honest with you, but it's what we have to work with. It works as follows. If we start here with oxaloacetate, and we take oxaloacetate and we add electrons, looks like the arrow's going the wrong way, but it's not. We add electrons to it, we create malate. That is, we're going backwards from the citric acid cycle reaction. Malate has 2 electrons. It has 2 more electrons than oxaloacetate has. Malate can be transported into the mitochondrial inner matrix. As it gets transported in, alpha-ketoglutarate gets transported out. Okay? As far as I'm concerned, that's not significant. What matters is malate's coming in. And malate is carrying electrons. And on in the matrix of the mitochondrion, look what happens. The reverse reaction occurs. NAD picks up the electrons from malate and produces oxaloacetate. We started with 1 NADH out here. We end up with 1 NADH in here. No loss. This means then that this system is much more efficient. It's not as fast, but it's very efficient because no energy is lost in this process. We have, in fact, 3 ATPs that are going to be realized by the pumping of protons from NADH. The rest of this figure, which looks very confusing, is simply balancing the equation. All right? I don't expect you're going to reproduce this figure. The critical part of this figure is what's happening right here. Malate is the shuttle that carries the electrons across. The oxaloacetate gets converted to aspartate. The aspartate gets transported out as glutamate gets transported in. Glutamate gets converted to alpha-ketoglutarate and goes up here. And the equation is balanced. Okay? The important thing isn't balancing the equation. The important thing is what's happening, how the electrons are moving. Yes? Is this happening during the citric acid cycle? The citric acid cycle is essentially always going on. Okay? So this is a reaction of the citric acid cycle. It's always there available to work. I shouldn't say always going on, but it's always available there to work. Yes? I'm sorry?
Can you please go again what's being transported in, what's out?
Malate is being transported in. Period. So the rest of it's balancing the equation. I don't care about balancing the equation. All right? This is a system that uses malate and aspartic acid to transport electrons. Okay? That keeps it simple for you, right? You don't want it simple?
No, I just want you already went through this, and I want to get it.
Okay. Malate's coming in, makes oxaloacetate. Oxaloacetate gets converted to aspartic acid. That's simply putting an amine group on there. Aspartic acid gets transported out as glutamate gets transported in. All right? Glutamate gets converted to alpha-ketoglutarate by simply taking an amine off. Remember the transaminase reaction I talked about last term? This is a transaminase reaction occurring right here. Okay? And alpha-ketoglutarate becomes the balancing thing to bring this in. Okay? You're not responsible for balancing the equation. Okay. Well, at this point, oh, by the way, we have to get ADP in there too, because ADP has to be in the mitochondrial inner matrix. We have to have ADP in there. It turns out that there's something called an Adp Atp Translocase. For every ADP brought in, 1 ATP is put out. Nice exchange, and it doesn't take any energy. Yes? Adp Atp Translocase. Okay? Now, at this point, we can use our knowledge, okay, about this system to understand one type of diet drug. And by the way, I think diet drugs are some of the most stupid inventions of mankind. Okay? So I'm not endorsing any diet drug. But I will tell you, and with this, what I'm going to tell you, I hope that you will see why diet drugs really are stupid ideas. There was a diet drug that was released about 100 years ago, and it was incredibly powerful. And it worked as follows. People took the diet drug before they went to bed, and magically in their sleep, fat got burned off, and they woke up in the morning, and they were less fat than they were the previous day. I hear laughter. That's exactly what they did. Okay? This is what you always hear on the internet. Melt those pounds off in your sleep, right? This diet drug did it, and it does it. It can do it. How does it work? Okay? Well, it works as follows. I got a piece of apple in my mouth. It works as follows. All right? It's a drug called 2,3-dinitrophenol, 2,3-DNP. Okay? I'm sorry? 2,4, yeah. I got 2,3 from 2,3-bisphosphate in my head. 2,4-dinitrophenol. Okay? Dinitrophenol, DNP is what it's called, all right, pokes holes in the mitochondrial inner membrane. Now, if you poke holes in the mitochondrial inner membrane, what's going to happen to the proton gradient? You're going to lose it. It's like poking holes in a dam. What's going to happen to the water behind it? Water's going to come out of the holes. It's not going to go through the turbine, and the turbine's not going to turn electricity, and you're not going to have much power. You poke holes in the mitochondrial inner membrane, the protons are going to leak back in. They're not going to go through the ATP synthase, and you're not going to produce ATP. Cells furiously try to make ATP by burning more and more of their fats and sugars and so forth, and people lose weight. And they lost weight. About 10% of the people died. Okay? Now, at this point, people in here are saying, so if we took just a little bit? Just a little bit? To which I say, how about a little bit of arsenic? It would accomplish exactly the same thing. Okay? You would lose weight. About 10% of you would die. And you probably wouldn't want to do it. Right? It worked beautifully. Okay? That's how the diet drug worked. Okay? It was messing with our metabolic control. Our metabolic control works as follows. The electron transport system is a necessary component for oxidative phosphorylation. If electron transport is not working, there will be no proton gradient. And if there's no proton gradient, there will be no oxidative phosphorylation. Oxidative phosphorylation is necessary for electron transport as well. Because if oxidative phosphorylation is not going on, and we have what's called tightly coupled mitochondria. Whenever you hear that phrase, it means intact mitochondrial inner membrane. When we have an intact mitochondrial inner membrane, all right, if we have no ADP, what happens to oxidative phosphorylation? The answer is it's going to stop. Because without ADP, the little gamma thing can't rotate. And if the gamma thing can't rotate, then protons can't come in. It blocks them. If protons can't come in, what happens to the proton gradient as electron transport continues to pump more and more protons out? The proton gradient gets very steep. And just like I talked about trying to push all that water upstairs, the more I push, the harder it is to get more to go up there. Similarly, the steeper the proton gradient, the less the electron transport system will run. The electron transport system will stop when you run out of ADP. When do you run out of ADP? You run out of ADP when you're sitting around, drinking beer, eating pizza, watching the tube. You're not burning ADP. Right? Hey, this makes sense. It makes really good sense. When I'm sitting around, drinking beer, watching the tube, and eating pizza, I'm not breathing heavily. Why am I not breathing heavily? Because my electron transport system is not running very well. I don't need very much oxygen. They're tightly coupled. The best evidence that you have tight coupling is that fact. And when the fire alarm goes off, and you jump up and you throw out your beer. No, you don't throw out your beer. You always carry your beer out with you. That's one of the rules. One of the rules of fires is always carry your beer with you. You might be able to put a fire out or something with it. You never know, right? You grab your beer and you go racing outside. You start burning ATP. And when you start burning ATP, you make ADP. And when you make ADP, what happens to the rotating gamma? It starts spinning. And when it starts spinning, what happens to the proton gradient? The proton gradient starts going down. And when the proton gradient starts going down, what happens to electron transport? It starts pumping. And when it starts pumping, what does it need? Oxygen. And you start breathing heavily. Now, tight coupling really makes very good sense. When you hear about respiratory control, I want you to immediately think tight coupling. And tight coupling means intact mitochondrial inner membrane. They're all the same thing. They all require the same thing. Okay? Let's think about those people that took the 2,4-dinitrophenol. Okay? The people that took the 2,4-dinitrophenol poked holes in their mitochondrial inner membrane. What happened to the proton gradient? Gradient went down. What happened to their electron transport? Nothing to stop electron transport. It's going to start because the proton gradient's down. It starts going, right? What happens to the use of oxygen? Goes up. That person, when they're sleeping, they're breathing heavily. Wow, I had the really restless sleep last night as I was burning off all these pounds. All right? The other thing that they're going to do is they're going to generate heat. Because when they are running electron transport very rapidly, what's happening to their metabolic processes? Citric acid cycle, for example. Up or down? Way up. Right? Citric acid cycle has a Big Bang reaction in it, right? Big Bang reaction produces heat. Right? It's also why when you're exercising, you get hot. You're producing those Big Bang reactions that are generating the heat. And that's why you get hot. Okay? Does this make sense? Start over here and then I'll get you. Yeah?
What happens to a mitochondria that has holes in it? For instance, like does it go away after a while?
What happens to a mitochondrion that has holes in its inner mitochondrial membrane? I'm not an expert in this field, but my prediction would be it would probably be destroyed because it would just burn itself out. Yep.
Why those 10% specifically die? What is the cause? The respiration problems or?
So why do those 10% of the people die? Okay. Well, imagine that you can't make any ATP. What's that? Okay, so he says, well, maybe they're burning fat. All right. Well, if they're burning fat, you're definitely going to burn fat, but you're still not going to make any ATP. No ATP is going to mean death.
That means they just overdose themselves, because usually probably drug was intended to have only some mitochondria to have holes, not all of them. I mean, not a significant amount.
He's making the argument about if I just took a little bit. Okay? No, he says, because you're overdosing yourself. Okay, if you're taking any of this stuff, you're overdosing yourself. Okay, so that's the point. That's the point. Yeah. So, yes, this is nasty stuff. It's not something to mess with. Okay? And if you think about it, whenever you see these internet things and so forth out there about magic diet pills, or even the legitimate claims of diet pills and so forth, they're really designed to mess with something that you probably don't want to mess with. Respiratory control is very important for you. You don't want to be fouling that system up. Yes? Is that what? Is it permanent when it goes in the mitochondrial membrane? Again, I'm not an expert in this area, but I think you would have a hard time getting it out. Yes?
How 90% survive? I'm just curious. If you do too much overdose, how does 90% survive?
How do 90% survive? I'm not a physician. Okay, and one of my very big principles is I don't give medical advice. So this borders on that, and I just can't and won't do that. So I'm sorry. Yes?
Maybe the wrong cells have the mitochondria function.
Maybe the wrong cells have their mitochondria. So you've got another approach here, right?
No, I'm just saying the reason I think that 90% survive
Yeah. I can't tell you. I can't say. I mean, my thought is that killing a lot of cells would not be a good idea. And this is a very common discussion in this class, by the way. It happens every year. So, I mean, because this really makes you think about stuff. It makes you think about how this system is being messed with 2,4-dinitrophenol. Okay? But I honestly can't tell you why 10%, because I don't know the answer to that question. Okay? But even if I knew the answer to that question, that would, as I said, would be bordering on medical advice, and I just don't want to give that, because I'm not a trained professional to do that. I'm a trained biochemist professional. That's different. I play a biochemist on TV. I made up a joke as I was coming in here today. Okay? You want to hear my new joke? What do you call a Ferrari that eats cheeseburgers? Carnivorous. I didn't say it was a long joke. Carnivorous. I thought that was, I liked it. But I always like my own jokes anyway. Okay. All right. So, we've talked about respiratory control with respect to diet drugs. Let's talk about it with respect to another of my favorite topics, and that's the photosynthetic fish. Okay. Photosynthetic fish, the idea is exactly the same. I shouldn't say exactly the same, but the idea is the same. We're going to use the mitochondrion to do something that we want, okay? But instead of destroying it, we're going to help it. All right? How does it work? Well, you might remember a few lectures ago, I talked about a protein called Bacterial Rhodopsin. Everybody remember that lecture? And you remember what the Bacterial Rhodopsin is doing? It's got the little leg, and it's got that vitamin A in there, and the vitamin A switches between cis and trans, like my leg, and it's kicking protons out of a bacterium. What if we took that bacterial rhodopsin out of the bacterium, and we put it into the mitochondrial inner membrane? And let's say we put it into something that was transparent, like a fish. Little fish in your aquarium. You've seen these little transparent guys, right? And let's say that we took that fish and we shined light on it. What is going to happen to the mitochondria of that fish? Well, is when I turn the light on, the bacterial rhodopsin is going to start kicking protons out of the mitochondrial inner membrane. We're going to create a proton gradient. And what are proton gradients good for? Making ATP. This photosynthetic fish now is capable of making ATP without eating anything. Solely on the basis of light, it's producing a gradient that's used to make ATP. Now, I'm using the term photosynthetic here a little bit differently than the sense in which we use it in plants, because in plants, photosynthesis not only includes the synthesis of ATP, but also the sucking of carbon dioxide from the atmosphere to make glucose. This fish won't do that part. But what this fish will do is make ATP extraordinarily efficiently. If we think about raising fish, okay, economically, fish will still have to have a carbon source. They would still have to have a carbon source so that they could grow and so forth. But they won't need nearly as much because most of the carbon source that we use is used for energy. These fish would only need carbon as a way, these fish would only use carbon as a way of making the structural components. There we go. The structural things that they need. Yeah?
Would you only be able to grow transparent fish, though?
Would you only be able to grow transparent fish? Actually, the transparent fish would probably be the most efficient fish that things that you would grow. But in principle, this could work in you. Put it in your skin cells. Now, if the only things that are exposed are your skin cells compared to the rest of the cells in your body, that's a pretty small percentage of the cells. That's why with a transparent fish, you're going to get many more cells that are going to be exposed to the light and have the mitochondrion exposed to light. Photosynthetic fish, I think, will work. There's your billion-dollar idea of the day. If anybody does it, I want rights. Yes, Kimberly. I was going to try it at one point. I've actually talked to some people who work on mitochondria, and they said, yeah, I think it'll work. But I have a lot of things going on, and I never got to do it, so I haven't done it. As far as I know, nobody's done it. I've mentioned it in the class now for 6 or 7 years, and I hope it's enough out there that people will actually hear about it and try it and let me know if it works or not. Yeah? Well, it's not a complicated idea. It's, you know, the idea is simply based on what Bacterial Rhodopsin does. You could have thought about it, but you probably didn't sit around. You're not like me, and you don't sit around thinking about Bacterial Rhodopsin. But yes? So he's got a very good question. Okay, if you expose the fish to a lot of light, would they just burn themselves up? Okay, and I think that the likelihood is that they might. So you might have to be very careful in how much light you expose them to. Imagine that you're going to create an enormous proton gradient, right? Are you going to short circuit mitochondria at some point? Yeah, you might. So but the beauty of this is the less light you have to use, the less energy it takes to make the fish. I need a low light fish, please. Right?
Do you usually do it by just taking this protein and incorporating it to another mitochondria, or you actually make this mitochondria to produce it? You just mess with DNA.
Yes. So his question is, how would I get the protein, the Bacterial Rhodopsin, into the mitochondria of the fish? The most likely way to do it would be to take the gene for the Bacterial Rhodopsin, incorporate it into either the bacterial genome, not bacterial, the cell's genome, or the mitochondria's genome. Okay? If you did it into the cell's genome, you would have to have what's called a bacterial targeting sequence that would point it as a destination to go to the mitochondria. But that's very doable.
So in this case, fish just can reuse the cells and still she'll be photosynthetic.
So the fish could use it and still be photosynthetic. It'd be kind of cool. Yeah. I want to see somebody do this. Somebody do it. Let me know if it works. What's that? Yeah.
So, like 10 minutes ago, you mentioned ATP translocase.
Yes.
I don't know, what are we supposed to know about it?
What did I say about it? So you're always supposed to know anything I say in class is fair game. So whatever I said about it is what you're responsible for. If you have a question about how it works, I can tell you, but what I said about it is what's important. Okay, all right, so maybe I'll clarify. The ATP Adp Translocase is simply moving ADP into the mitochondrial inner matrix. I'm sorry if I didn't say that. Sorry. Okay. So, and that is important because, again, if without ADP, what's going to happen? Nothing good. Can't run this without ADP. We also want to take that ATP that we've made and move it out. Okay? Now I see several hands. Let me go back up here first, and then I'll come back down here. Yes. So because it's a vertebrate, there may be logistical hurdles of doing this. His question is, would it be better to do a photosynthetic worm? I think with any of these, it's the principle is one of just demonstrating that it works. But I agree with you. You have to take those things into consideration, definitely. So was that? Yeah. Vertebrates would be problematic. Well, there are genetically modified vertebrates that are out there. I mean, there are genetically modified fish right now, so it's doable. But you would have to go through the approval process to do something like that. Yeah. So it's not a trivial consideration. Yes.
If you were going to use this process, would the fish still have to uptake organic matter to account for minerals?
Yeah. So his question is, would the fish still have to take up things? And that's what I'm talking about, carbon source and so forth. Yes, it would have to uptake something as a carbon source because it's not pulling carbon dioxide out of the atmosphere. Plants are doing that. So there's that consideration. Yes? Okay, so good question. His question is the ADP ATP translocase doesn't use energy to accomplish it. Does it use its concentration gradient to make this process happen? What do you think? How many yays? How many nays? No nays. The majority has it. Yeah. So the only thing available is a concentration gradient. So again, this is going to be driven by high ADP levels inside, high ATP levels outside. You're going to swap. So yeah, that's exactly the driving force. Okay. I kind of got off on a tangent there, didn't I? Okay. Any other questions? Those are good questions. Okay, well, let's go through and just sort of finish up the control of this, and then I've got a song we'll finish with for the day. So respiratory control, no, I don't like that. Respiratory control is limited by the concentrations of ADP, oxygen, and NADH. All right? Those three things really control respiratory control. If you have high NADH, okay, and you have low oxygen, what's going to happen? Think about it. That's a good exam question. What's going to happen? If I've got a cell that's got high NADH and low oxygen, what's happening in that cell? The cell is not going through electron transport. Why is it not going through electron transport? Oxygen is probably limiting, so oxygen has stopped the process, which backed up electron transport, which backed up the dumping of electrons from NADH. What else is not going on in the cell? We don't know about ATP synthesis. Can't tell you from what I've told you there. So ATP synthesis is out. David? Citric acid cycle is going to back up. Citric acid cycle needs NAD. Absolutely. Okay? What's probably happening with this cell? Or in the individual that has this cell? What is this individual probably doing? What's that? Well, not necessarily. He said producing lactic acid. It could be. Fermentation? Possibly, but that's not the most likely explanation. What's that? They're relaxing. Right? Now, it's possible they're suffocating. It's possible that's how you get low oxygen there, right? And if you were suffocating, you would have high levels of NADH because you wouldn't have electron transport going. Let's say we added to that fact that we had high levels of ADP. Now tell me what's going on. So we have high NADH, we have low levels of oxygen, and we have high levels of ADP. Tell me what that person is doing. They are either exercising very heavily and or they're suffocating. Okay? Because they're using as much ATP as they can, but they can't get the other processes going, meaning that the oxygen is limiting. Oxygen can be limiting under two circumstances. One, if they're exercising faster than the blood can deliver the oxygen, in which case heavy exercise makes sense. Or two, they've got a bag on their head and they've pulled it tight. Okay? Makes sense? Let's imagine that we had this person who has this circumstance, and we just say, okay, we're taking the bag off your head. Now you can go ahead and you can breathe normally and everything. And we kind of let them do their thing, and then we say, okay, I'll tell you what. How about trying some cyanide? Tell me what's going to happen to these molecules. What's going to happen to the level of NADH? Going to go up, right? Why is it going to go up? Electron transport chain is blocked at complex 4, right? Electron transport chain gets blocked, NADH can't dump off its electrons. What happens to oxygen consumption? Oxygen consumption goes down, which means oxygen concentration goes up. Okay? What happens to levels of ADP? It's going to go up. Why is it going to go up? Because they can't make ATP. If you can't make ATP, you're going to burn up what ATP you have, and your ADP levels are going to increase, but it's not going to get converted back into ATP. Respiratory control is pretty cool. Do you understand how these interconnections make this possible? Let's imagine that we had this person, we said, okay, well, here's your cyanide. You've taken your cyanide, all right? And then you say, oh my God, I didn't mean to give you cyanide. I gave you cyanide. I'm going to give you some 2,4-DNP to take care of it. Now what's going to happen? What's going to happen to the levels of NADH? So the cyanide's still there. So NADH levels are actually going to go down. Okay? Because electron transport chain is going to start, right? So electron transport chain starts, NADH levels go down, right? What happens to the citric acid cycle? Takes off, because as NAD levels go up, citric acid cycle is able to start, right? What happens to oxygen consumption? It's going to go up or it's going to go down? It's not going to change at all, because we still have the block at complex 4. So if it got to that higher level before, it's going to stay right where it is, because we can't transport electrons onto oxygen. Oxygen is going to stay the same. What's going to happen to ADP levels? Again, they're going to be low because can't make any more ATP. So the only thing we've changed in adding the 2,4-dinitrophenol is we started burning NADH, we started converting NADH into NAD, and we started the citric acid cycle, but we did not cure the problem. So now you guys, there's a piece of medical advice. 2,4-dinitrophenol will not cure cyanide poisoning. Yes, question? Okay. Cyanide blocks complex 4.
So, while you're changing the flow of electrons will flow back into the mitochondria,
It's the flow of protons. He said the electrons flow back into the mitochondria, and it's the protons that are flowing back into the mitochondria. That's right. So NAD concentrations go up, NADH concentrations go down. Yes, over here. If cyanide, if the electron transport chain is blocked by cyanide, how is the electron transport chain still running? Okay. So, okay. So, let's see. So there are ways of short-circuiting that complex, which is basically what's going to happen here. But your point is a good one. If you can't donate electrons to oxygen, you are going to gum things up, right? So in essence, you're not going to generate things going a lot more as a result of adding the 2,4-dinophenols. It's a very good question, very good observation. David?
So, complex 4 is blocked by complex 1 and 2, so we complex electrons
They will, so in other words, what's happened here is you've relieved, this also is related to her question here. Okay? So what you've done is you've relieved the block which you had as a result of the proton gradient. All right? That contributes to the overall stop of the process. Protons come in, all right? You can still squeeze more out until you gum up the system, and you can't add any more because the chain itself is blocked. So that's her question. That once the chain is blocked, is it going to start again? The answer is no. If the block was the proton gradient, when the protons come back in, that will pump more protons out until the whole system gums up. Do you see what I'm saying?
Wait, so since there are holes in the mitochondrial membrane,
The protons are flowing back in. That's right. But her point is that the electrons still have to have some place to go. Okay? So without a place to go, those electrons are going to gum up the system. So she's right. Well, ultimately, they go to oxygen. If they can't go to oxygen, then the system's going to gum up. That's what I'm saying. Okay? What's that? Well, there'll be second 4, then there'll be second 3, there'll be second 2, there'll be second 1. It backs the whole system up, right? Like a traffic jam.
so it only starts to burn up the NADH to a point.
That's correct. That's correct. It burns up the NADH to a point. It's not going to go willy-nilly crazy. Very good questions. Very good questions. This is good to think about. Yes, Omar. Why is ADP going down? Yeah. In the first system, we talked about ADP going down because no ATP is being made. In the second case, ATP is still not being made. So it's going to go down and down until you run out completely out of ATP. Yeah, question. Yeah, is there any way to repair the mitochondrial membrane after it's been damaged? And the answer is, I don't know. My suspicion is once you've damaged it, probably the mitochondria itself would be replaced, but I don't know the answer to that question. Yes? What happens to oxygen in the case of cyanide poisoning? Well, you tell me. What's oxygen used for? Okay, so what's happening to electron transport? It's off. So what happens to oxygen use? It goes down. If you stop using oxygen, what happens to your oxygen concentration? It goes up, right? So I want you to learn to think about these things the way I'm describing them to you. All these interconnections, you change one, you affect the next one, and so forth. Okay? These are very good exercises to think about. Okay. We've had an unusual weather year this year. And I have a song I usually play at about this time because it's usually raining cats and dogs. And this year it's not raining cats and dogs. So, but nonetheless, I'm going to play the song because maybe it'll cause it to start raining cats and dogs again. And I hope you'll join me. Sky above's not clear again, and the sun has disappeared again. Crappy days are here again. Rain is falling from the sky. I wish I knew the reason why. Guess I'll have to wait until July for the weather to be dry. All right, see you guys on Friday.